Proteus. It belongs to the tribe Proteae, and the tribe Proteae can be divided into 3 genera. The most important one is Proteus. You also have Morganella and Providentia. So these are the 3 genera that you have to remember when it comes to the tribe Proteae. Now coming to Protease, there are 5 points that you have to remember about Protease, which is very, very important. Now the first point is Protease is pleomorphic. So this is in fact the reason why it is considered to be called as Protease. Now the other thing is it shows a phenomenon which is known as swarming. The next one is it has an enzyme which is known as phenylalanine deaminase. So it has this particular enzyme which is in fact the basis for PPA test. Now the next one is it shows a phenomenon which is known as Diene's phenomenon, and the last one is its role in Weil-Felix test. So you have to understand that it has a role in Weil-Felix test. So when it comes to Protease, these are the 5 questions that are usually asked about it. And coming to the others, they are all grouped together in one particular tribe for certain similarities that they share. Like Proteus, Morganella and Providentia, all of them are motile. So they are all motile. And they all have a peculiar smell which can be known as seminal smell or fishy odor. So this is the typical smell or the odor that you have pertaining to it. Now these are normally isolated from various areas and these can be seen as saprophytes, but many a times there is a risk that these can also turn into nosocomial infection causing organisms. So these are responsible for nosocomial infections. Now the last one is that all of these 3 organisms produce the enzyme phenylalanine deaminase. So it produces phenylalanine deaminase and they are all positive for it. So whenever you are testing an organism or trying to differentiate it from the various other organisms of Enterobacteriaceae, you can actually do it with the help of a PPA test. So PPA test is positive for all the 3 organisms, but the differentiating feature that you can see between Protease from the others is, though all of them are motile, Morganella does not show swarming. So there is no swarming for Morganella, though it is considered to be motile. You do not see swarming even with Providentia. So these are the various important differentiating points in fact between Protease, Morganella and Providentia. Now Morganella morgagni is in fact only one species of importance for humans, and most of the times these are infections that occur mostly causing wound infections, and these are nosocomial infections, not normally seen, because these can also be seen in as these are normally seen as saprophytes. Now clinical importance. Now what is the importance of Protease? So we're going to discuss about Protease now. And what is the clinical importance of Protease? Now number 1, you have to remember that these are saprophytes. So these can be seen in frequently present in various areas. So whenever you have sewage or when you have decomposing material, you are able to isolate Protease. Now the other thing is it produces opportunistic infections. It is capable of producing opportunistic infections in certain conditions, like urinary tract infections or wound infections, or it can cause septicemia. So many of these things can be caused by Protease. Now the other thing is these are normally isolated from moist areas. So moist areas of the body. So wherever you have moist areas, you can be able to isolate Protease. Now the last important point that you see is Protease is associated with struvite stones. So if you have come across struvite stones in the kidneys or in the bladder mostly, that's where you see your struvite stones. And why does this happen? Now you have to understand as to why this basically happens. Now first is Protease is a strong urease positive organism. So it produces urease enzyme. So Protease produces urease enzyme, which in turn what it does is it breaks down urea to form ammonia. So because of which the urine becomes alkaline, and as a result of urine becoming alkaline, there is a predisposition for phosphate being deposited. So you see more amount of phosphate being deposited. So deposition of phosphate happens. This leads to caliculi, which can also be renal caliculi or can also be your bladder caliculi. So this is what you normally see. So Protease is responsible for stoid stones seen in bladder and also is responsible for renal caliculi. So this is because Protease produces urease enzyme. So urease enzyme breaks down urea into ammonia, and this is responsible for making urine alkaline. Because of the alkalinity of the urine, there is increased deposition of phosphate, and this is responsible for formation of renal caliculi. So these are the important clinical features that you have to remember. That is, whenever you are isolating Protease from a culture, history becomes very, very important, because these can normally be isolated from various areas in the body, especially the moist areas of the skin. So history pertaining to what the condition is very important when you are isolating organisms. Now coming to swarming. So you can see swarming here, and this is nothing but Protease which is grown on blood agar. So swarming of Protease can be seen in two forms. So the first form is continuous swarming and the other is discontinuous swarming. So this can be seen continuously or discontinuously. So swarming is an important feature to remember pertaining to Protease, and like I had already told you, this is in fact something which is not seen with Morganella or Providentia, though they belong to the same tribe as Protea. So this is the differentiating feature. So you take a blood agar plate and you put a small colony. You tend to see concentric circles most of the times, and this is nothing but what is understood to be as swarming. Now what typically happens in swarming is in the edge of the colonies that you have, you tend to have vegetative cells which are not very highly flagellated. So these again convert into multinucleated flagellated cells, and and then they become swarm cells, and these are responsible for the phenomenon of swarming. So whenever you see a plate, it's very, very easy to identify that this could be a case of Protease. Now the swarm cells are usually elongated cells, and these in fact move in concentric circles. So that is how your swarming growth happens in your blood agar. So if you've noticed here, the colony was inoculated at this particular point, but you can see concentric circles that have moved towards the outside of the medium. So this is what is known as swarming, and swarm cells have the tendency to move towards uninoculated part of the media. So that is in fact how it keeps moving. So swarming is in fact something which is demonstrated only by Protease, very typically seen by them. Now there are few other organisms. So there are other organisms which can also show swarming. So unlike Protease belonging to this group, there are other organisms belonging to other groups which do show swarming. Now some of them are Serratia, Vibrio parahemolyticus, and Clostridium tetani. So these are the various ones that you can remember as the organisms which cause or which show swarming in a culture plate. So Serratia, Vibrio parahemolyticus and Clostridium tetani are all the organisms which show swarming. Now coming to the problem with swarming. So whenever you have a culture which has multiple growth, so there are multiple organisms which are growing. Now the problem with Proteus is it tends to cover the entire blood agar plate. So it's very, very difficult to isolate the other organism when it is growing along with Protease. So what can be done about it? So what needs to be done about Protease? Now coming to this, there are few other things that can be done to inhibit swarming of Protease on blood agar and other media. Coming to morphology, it's a gram-negative bacilli, and like I told you, it is most of the times pleomorphic. So it has multiple shapes that it shows, but it is mainly gram-negative bacilli. Now swarming is seen, and like I told you, this is because of the swarm cells. So swarm cells are responsible for swarming, and these are seen at in fact these are the ones which are seen at the edge of the colony. That is, the vegetative cells become highly flagellated swarm cells. Now how do you inhibit swarming? So I have shown you swarming. Now you would want to inhibit it for reasons that you would want to isolate the other organisms in the culture. So it is done by increasing the percentage of agar from 2 to 6%. You can add some chemicals like boric acid and chloral hydrate. Now you can culture it on other media like McConkey and phenylpyroic acid agar. Now the advantage of this is a differential medium. So when you have McConkey agar, it's a differential medium. It inhibits swarming because of the excess amount of bile salts, and it also shows that it is a non-lactose fermenting colony. So you will be able to see pale colonies on McConkey agar. So this is something that you will be able to see. Now you have other media like phenylpyroic acid agar, but the thing that is very specific with the media, or in fact something that we all know in the laboratory, is the smell of Protease. So it is a fishy seminal smell that you can feel even at one corner of the lab. You'd be able to smell it when they open a plate of Protease. So that is the typical smell that you have when it comes to Protease. So this again is swarming in your blood agar plate. Now coming to Diene's phenomenon, like I told you, this is another important concept that you have to remember. Now what is Diene's phenomenon? So here what you're doing is you took a blood agar plate, and here you have inoculated two different strains. So you've inoculated one colony here and the other colony here. So these are two different strains in two different areas. So whenever you do not know what the strains are, or if the strains are the similar ones, what you do is you inoculate it on two different areas, and when the swarming of two strains merge incompletely, it shows that the strains are different. Now if the swarming of the two strains merge completely, it shows that they belong to the same strains. So in this particular picture, you would be able to see that there is a small gap here. It's a narrow visible furrow that you can see. That is that they have not completely merged in here. So this phenomenon is known as Diene's phenomenon of Protease vulgaris. So let me repeat. So Diene's phenomenon of Protease is when you have two strains of Protease and you would want to see if they are similar, you inoculate it on two different parts of your blood agar plate. Now what you see is, if suppose they merge incompletely, it shows that they belong to two different strains. But if the swarming merges completely, then it shows that they belong to the same strain. So this line of demarcation is the one that helps you to know if this is belonging to the same strain or a different strain altogether. So this is known as Diene's phenomenon of Protease. Very important to know exam point of view. Now coming to the next test, which is known as PPA test. So this is in fact the one which is needed to test for or differentiate Protease and Morganella and Providentia from the other different tribes that you have. So in fact the entire Protea can be differentiated from the other tribes of Enterobacteriaceae. And how do you differentiate it is with this particular test, which is known as PPA test. So PPA test is a test, in fact, which tests for the enzyme that I had been telling you, which is known as phenylalanine deaminase. So like I told you, Protease, Morganella and Providentia produce phenylalanine deaminase, and this breaks down, this enzyme in fact breaks down phenylalanine to phenylpyroic acid. So this is in fact the basis for your PPA test. So your phenylalanine is being broken down to phenyl pyruvic acid. So this is something that you normally see, and this happens because of the presence of your phenylalanine deaminase. So this in fact is the enzyme that is responsible for this particular test. So once you have phenyl pyruvic acid along with ammonia which is produced, now when you want to demonstrate this, what do you do here? So to the media you add 10% ferric chloride. So this is in fact added, and this is responsible for the green color. Okay, so the green color which is considered to be positive is after you add ferric chloride, and this ferric ions change the media to acidic and give this particular color which is green color. So if you can see a media which is present here, so here again they have added ferric chloride here, but since there is no phenyl pyruvic acid which has been produced in this particular test tube, you tend to report this as PPA test negative. But here what happened is, like you notice here, this is green in color, and this is because your phenyl pyruvic acid which was produced was tested again with ferric chloride, and this gave the typical green color, and hence this is considered to be PPA test positive. So this is in fact the differentiating feature for Morganella, Providentia and Protease. All three of them are PPA test positive and can be differentiated from the other organisms of Enterobacteriaceae. Now Protease antigen interactions. Now this is in fact one of the tests that you have to remember, which is known as wheel Felix test. So there are certain Protease species which are non-motile. Now we've always been talking about Protease to be motile organisms showing swarming, but there are few non-motile Protease species. So these are the ones which are used for your wheel Felix test. So OX19, 2 and OXK in fact are the ones that are used for your wheel Felix test. And what do you see here? So OX2 and OX19 are the ones which you get from Protease vulgaris. So there are two important species to remember. One is Protease vulgaris and the other is known as Protease mirabilis. So these are the two important ones to remember. So this particular OX19 and OX2 is derived from Protease vulgaris, whereas the other is taken from Protease mirabilis. So these are the things that you have to remember. And this is basically an agglutination test. And what do you see here? So Protease antigens in fact have the tendency to cross-react with certain antigens of rickettsial species. So Protease antigens are used to detect heterophile antigens. So whenever a patient is suffering from rickettsial disease, you use your Protease antigens to detect the heterophile antigens. So this acts in detecting the heterophile antibodies rather. So you detect the heterophile antibodies which are present in your in the sera of a patient who is suffering from rickettsial disease. So this is in fact the basis for wheel Felix test. To repeat, there are certain strains of Protease which are non-motile. So these have a similar antigen which cross-reacts with the other antigens of rickettsia. Now, because of this, you tend to use this for an agglutination test, which is known as wheel Felix test. And Protease antigens are used to detect the heterophile antibodies which are produced in a person who is suffering from rickettsial disease. So this is the basis for wheel Felix test. So wheel Felix test is an heterophile agglutination test. Now OXK is strongly positive for scrub typhus. Now Protease is associated with chronic UTI. Now like I told you, because of formation of struvite stones. And this is also capable of forming your staghorn caliculi. So this can in fact lead to renal failure. Now this is mainly because of increased production of urease enzyme. So this is something that you have to remember. So you tend to see a lot of struvite stones. And rickettsial disease, and like I told you, this is in fact where we are trying to do. So day 6 onwards, in a patient suffering from rickettsial disease, you tend to do a wheel-Felix test, and this is an heterophile agglutination test because of a similar cross reaction between somatic antigens of Protease and certain antigens of rickettsia. So that is in fact is the basis for wheel-Felix test. So these are the important things that you have to remember. To summarize, there are 5 important points about Protease that you should know. It produces an enzyme which is known as phenylalanine deaminase. This is in fact the basis for your PPA test. Second, it shows swarming, and that is because of swarm cells. Now the third one is what is known as Diene's phenomenon, especially when you are trying to differentiate 2 different strains. So there is a line of demarcation if they belong to different strains. Fourth, it shows pleomorphism, and though it is a gram-negative bacilli, it shows pleomorphism. And the last important point is wheel-Felix test, which is an heterophile agglutination test that is used for diagnosis of rickettsial diseases.